vogliamo stare qua.
First of all, next Wednesday we don't have a class, right? Next Wednesday. And next Wednesday, yes.
Wait,
You're not today. Your time to party. To get wise. But Sunday there is the Gatsby meeting. And it's going to be here. Therefore, our class is going to be in the 504. 504, I think. 50 something. I will post it on the Moodle. And also I have the papers for the auditory and the papers for the last session. Have been decided. So I'll post them up. And basically this is the last הישורת האחרונה. The course.
Good.
Right? Last course. Okay. So.
Okay.
Shoot.
Okay. I'm going to talk about the paper Random Convergence of Olfactory Inputs in the Drosophila Mushroom Body by Caron and others. And we're going to start with a short reminder about the basic structure of the olfactory system in the Drosophila. And olfactory perception in the Drosophila starts in the antenna. That's where the primary olfactory receptors are. So an odorant binds to an ensemble of olfactory receptor neurons, and this results in the activation of the unique and fixed combination of glomeruli in the antennal lobe. And then projection neurons send dendrites into a single glomerulus and send their axons to two distinct regions, the lateral horn and the mushroom body. The circuitry of the lateral horn is relatively fixed, so it is thought to be connected to innate behavior, and the mushroom body, people think that it is involved in olfactory learning and memory. And now we'll take a closer look at the mushroom body, which is the region that we're interested in this paper. So like we said, projection neurons send their axons to the mushroom body. And their axons terminate in large butons, which is what we see here. And the intrinsic neurons of the mushroom body are called canyon cells. Their somas are here at the calyx, and they each have a few dendritic claws, these green things. And each dendritic claw receives information from a single projection neuron buton, but each buton can be connected to dendritic claws of multiple canyon cells. And this is how we get this special structure that is called mycoglomerulus. And it's basically just a projection neuron buton and the dendritic claws from different canyon cells around it. Okay. Now that we have a little bit of background, we're going to talk about this paper. The aim of the paper is to characterize the glomerular origin of the projection neurons that converge onto individual canyon cells. So in other words, they're asking, is there some rule that determines which glomeruli each canyon cell receives information from? And from the title, we can guess that they're going to say no. And we know from previous electrophysiological and imaging studies that when odorants bind to receptors, it activates a sparse subpopulation of canyon cells that are distributed across the mushroom body. And this leads us to two options. Option A, individual canyon cells receive information from preferential combinations of glomeruli, or option B, the inputs are not structured in any way and they receive information from random combinations of glomeruli. So in this paper, we're going to try to show which one of these options is true. And how did they actually do it? So in order to determine which developed an anatomic tracing technique, which is one of the most impressive aspects of this paper. And they did it while exploiting the special structure of the projection neuron canyon cell synapse, namely the fact that each canyon cell claw receives information from a single projection neuron that receives information from a single glomerulus. So this allowed them to trace back each claw to its glomerular origin. And then they perform a series of statistical analysis and trying to detect structure in these connections. The method. The tracing was done in transgenic mice, expressing PAGFP in all of their neurons. PAGFP stands for photoactivatable green fluorescent protein. So it's a mutant from GFP that that is able to increase in fluorescence over 100-fold when it's after activation using light in a specific wavelength. So, like we see in the picture, we are able to activate a single cell by shedding light on it, and then we can trace it very easily. So these slides express PAGFP under the control of synaptobevin or fruitless. Synaptobevin is a broadly expressed neuronal gene in the fruit fly, and fruitless is expressed specifically in one type of canyon cell, gamma cell. Now to the actual tracing. This is a canyon cell that was labeled by consecutive exposures to light and using a 2-photon microscope. And we see that it has a few dendritic claws, and we're going to focus on one. So this claw outlines a microglomerulus. At the center, there's actually a projection neuron button, and around it, the claws of other canyon cells. So what they did is they placed an electrode loaded with red dye at the center of this structure, and then they electrophoreted the dye into the projection neuron button. And then retrograde transfer labeled the projection neuron and the associated glomerulus. So in the big picture we can see up there it's the canyon cell, and this is the projection neuron in red, and you can see its button up there too, and this is the associated glomerulus that was also labeled. So this is pretty impressive, I think. The canyon cells were chosen randomly, and one canyon cell was used per fly. And then they performed this process sequentially for the different claws of the canyon cell. Okay, but this is an anatomic tracing method, so how do we know that these connections have any functional meaning? So in order to prove that these connections are functional, they used a combination of calcium imaging and dye electrophoration. And what they did is in flies expressing GSAMP3, they stimulated electrically one glomerulus. That resulted in the activation of a few microglomeruli in the calyx, and this is what we see here. This is the calcium imaging signal. And then they chose one and applied the same method as before with the dye electrophoration, and they see that the glomerulus that is labeled in red is indeed the glomerulus that they stimulated. So we know that this method allows us to detect functional connections between canyon cells and glomeruli. Using this method, they were able to obtain information from 665 claws from 200 canyon cells. These canyon cells receive information from 49 out of 51 glomeruli, so 2 glomeruli are not represented in this data at all. And some of them also receive information from pseudoglomeruli, which are thermosensing centers in the brain of the fly that receive input from distinct receptors in the antenna. And this is the connectivity matrix that describes all of the connections show is the canyon cell. A red bar means that the canyon cell and the glomerulus are connected once, and a yellow bar means that they're connected twice.
And just what about the top axis? Is it the same thing?
Yes, so the upper panel is like a summation of all of these columns. And so here we just see all of the connections distributed by glomerulus. These are the names of all of the glomeruli. And an important thing to notice is that this distribution is not uniform. We see that DA1 and DC3 glomeruli are responsible for about 5% of the connections each, which is more than any of the other glomeruli. So this non-uniform distribution already introduces some inherent structure to the data. Okay, now that they have mapped all of the connections, they can start asking some questions. So they perform a series of statistical analysis, and they ask the following questions. Do canyon cells receive preferential input from a single glomerulus, pairs, trios, or quartets of glomeruli, and preferential categories of glomeruli, and we'll talk about what these categories are a little later. And then they also perform an unbiased search for structure. And before we start looking at the actual results, just one important comment about the analysis. Okay, so after we perform the appropriate analysis to answer each of these questions, they still need to know whether the frequency of convergence of input upon individual canyon cells is above or below chance expectations. Meaning, does it come from this inherent structure that comes from the non-uniform distribution that we just talked about, or does it come from some other form of internal structure? So how did they dissociate these two factors? What they did is they shuffled all of the connections between canyon cells and glomeruli while maintaining the same number of connections per glomerulus. So in this way, they maintain the non-uniform distribution, but they eliminate any other non-random patterns of inputs onto canyon cells. And then they repeat the analysis, and if we get very similar results, we know that these results don't come from some interesting internal structure. So all of the analysis that we're going to see was performed on both the experimental data and 1,000 sets of shuffle data. The first question is, do canyon cells receive preferential information from a single glomerulus? So if we know that a canyon cell is connected to a given glomerulus, it is more likely to have more connections with this glomerulus. So out of 200 canyon cells, only 11 formed double connections with any glomeruli. And we can see that there are 9 glomeruli that form double connections with one canyon cell, and 2 that form double connections with 2 canyon cells. The gray bars are for the experimental data, and the black dots are for the shuffle data set. So they also say that there is no significant difference between the results for these two data sets. So the conclusion is that canyon cells integrate data from multiple glomeruli rather than receiving preferential data from a single glomerulus. And then they ask the same question for pairs, trios, and quartets of glomeruli. And here we only see the results from pairs, but for trios and quartets it was very similar. And this figure is exactly like the previous one, and again we see no significant difference between the experimental data and the shuffle data. So here too, they don't detect any interesting structure. Okay, so until now we looked at small random groups of glomeruli, but glomeruli can also be grouped according to biological properties of their projection neurons or their associated olfactory receptor neurons. For example, each olfactory receptor neuron expresses usually one type of receptor, olfactors are called sensilla, and they can be divided into three major groups. Here you're supposed to be able to see the different kinds of sensilla. I don't see any difference, but anyway, there are three: basiconic, coeloconic, and trichoid. So, okay, and then the olfactory receptor neuron, each type of receptor neuron innervates a subset of glomeruli, and then the glomeruli can be divided into groups according to their sensular origin. What they did here is they took all of the connections between canyon cells and glomeruli and divided them into groups according to the sensular origin of the glomerulus that is involved in this connection. So B, C, and T are the different types of sensilla, and U stands for uncharacterized. And then they took all of the canyon cells that form connections with basiconic glomeruli and took all of the remaining connections that they have and divided them in the same way. And then they repeated this for the other two types of sensilla. Okay? And we see two things. First of all, again, no significant difference between experimental and shuffle data. And another thing that we see is that these four figures look kind of the same. And we would expect if canyon cells indeed receive preferential input from glomeruli based on their sensular origin, we would expect that here when we're only looking at canyon cells that are connected to basiconic glomeruli, we want to see more basiconic connections or some sort of change, and we don't see that. And this is just one example, but they also looked at, they also created categorizations of glomeruli based on the morphology of the projection neurons, their developmental origin, odor response patterns, and other things, and they don't see any interesting structure anywhere. So the conclusion is that canyon cells do not receive preferential input from glomeruli based on their biological properties. Okay, and now they performed an unbiased search for structures using PCA, principal component analysis. Now in just very few words what PCA is, it's a statistical procedure that allows us to extract from the data a set of linearly uncorrelated variables that account for the variability in the data. And these variables are called principal components. They are arranged according to their variance. So the first principal component has the largest possible variance, and it accounts for, it contributes the most to the variability in the data. And here they performed PCA on this connectivity matrix. So it's a 200 by 53 matrix, and they got 53 different principal components for the 53 glomeruli. And here for each component we can see the percentage of the variance in the data that it is responsible for. The red dots are the experimental data, and the black dots are the shuffle data. And we see that the curves are very similar, so using this method, again, they don't see any structure. So their conclusion is that the data are consistent with a model in which each canyon cell receives information from a randomly chosen set of glomerular projections to the mushroom brain. And this makes sense in light of the role that the mushroom brain has in learning and memory. Because if the connections are indeed random, it means that the same odor will activate a different combination of canyon cells in the brains of different flies, but in the same fly it will consistently activate the same ensemble of canyon cells. And a structure like that is most likely to come from an activity-dependent plasticity. So it enables the fly to assign meaning to a big variety of unpredictable stimuli that it's going to encounter throughout its life. So the randomness in the system maximizes the ability of the fly to respond appropriately to different olfactory experiences. only allows for a single canyon cell per fly to be traced. So this means that there could be some structure in the system that we are missing in this process. But this would happen if there is a determined input to each of the 2,000 canyon cells in the brain of each fly, and this doesn't seem very likely. Another point is that this analysis was done on 200 canyon cells, and they were taken from 200 flies, and they each have 2,000 canyon cells, so it is possible that there is some subset of canyon cells that do receive preferential input from glomeruli. And another point is maybe a little obvious, but so if we go back to the title of the paper and ask, did they prove that the convergence of input is random? And for me, the answer is no. So they looked for structures in several ways that seem very reasonable, and I think it's really impressive, and they did a very good job. But still, it is theoretically possible that canyon cells receive preferential information from groups of 5 glomeruli, or based on some biological property that we're not aware of yet. So just because we haven't found any structure doesn't mean that it doesn't exist. But that's like a logical problem with proving randomness. Okay, so now to my personal view. I really liked this paper, and I thought it was elegant and cool. And one thing that I liked is the idea of looking at randomness in the system. So most of the time, for obvious reasons, we look for a structure and for what is common. But I think probably many of the processes that determine the way that the brain operates and develops have a very strong stochastic random component to them. And this reminded me a little bit of what Yves Mendel talked about when he gave a seminar here about the way that different mechanisms can give rise to very similar behavior. So I think it's interesting and important to remember to look at this randomness and to these differences between individuals, and there is something to be learned from that too. And another thing is, so we are mostly interested in the human brain, and this paper is about the olfactory bulb of the Drosophila, which is probably very different from ours. But still, I think they use this apparently more simple system to make a general point that can be applied for different species. And so, that's it. Any questions?
In the previous slide, in the second point, you mentioned that they only use the subset of very small. Was it possible that so the KT, the
Canyon cells?
canyon cells, thank you. Is it possible that sorry, I had a question. I will remember it in a minute.
Okay.
Did they look at differences between individuals, or is it random? So
Yeah, it's just this technique. It's very technically difficult, and they couldn't look at more. What do you mean by differences
if,
between
for
individuals?
example, there is a biological property that explains the connection, so we see the same in different individuals. But it's random, so we see.
Yeah, that's the point. That you only have one canyon cell per fly. So I'm not sure if I understand how to go.
So, do you think there is a better way to make the proof of randomness more believable?
No.
So,
Did they provide any other systems in the brain, maybe not in Drosophila, but in that there are randomness?
They don't talk about any other system. That's right. They only talk about this.
I remember they use the system of checking randomness for the entire population of canyon cells that they extracted. Would it be possible to use the same system for individual canyon cells?
For
With maybe less integrity of data, but still...
For different canyon cells in the same individual?
No,
You
no,
mean?
for a single canyon cell. Canyon cell.
And what do you mean?
That maybe some structure of the data gets lost in the
They
entire
only have
population.
like three inputs to okay, so canyon cells have more than three dendritic claws normally, but they with using this system they could only trace back 3 or 4 claws per canyon cell. So you mean like doing the analysis on with 4 inputs?
Yes, I understand that it's too small a data.
I don't know enough statistics to say, but I doesn't seem, or I don't know, maybe it is, but to me it doesn't seem. Okay?
Cool. Thank you very much. So anything, maybe we'll go get a microphone. Is that okay? Because for my hairstyle, I might need a boost. אז אולי מישהו יכול לעזור לי להביא, אני אזוז לשירותים. אני מביא לא רק ההרסל. הנה, לייזר פה. נסעתי להביא לייזר.
איזה סיפור הוא נלמד?
שהוא כתב לי כשדיברתי שהרזולוציה לא טובה או משהו כזה, ובאמת אמרתי, אני לא בדקתי,
אני לא בדקתי לי עדיין, אני גם לא מצליח להגיש מחשב שלי את הקוד ליד מה שלא לקרוא את זה. זהו, גם את שלי לא קראתי מה שכתבתי.
כתבתי לך וזהו.
אני כתבתי
יש פה פוינטר? יש.
זהו, חיפשתי אותו. הוא היה פה בכלל.
כן, אני יודע. אני חשבתי מה אפשר להגיד את זה, לא חשבתי את זה. הוא לא אומר, אפשר לך. לפתור, אתה יכול להגיד כמה אפשר תשובה לו.
אמרתי, משהו שתלמיד בקלינית, אני לא בטוח איך זה מסתכל.
זהו, זה נורא שם.
לא, וגם היום?
Just one point about this. There's a lot of debate now and a lot of discussion about specificity and randomness and sensitivity. There's a lot of money going into building the very precise connectivity matrices. And this is very. I think you did the dense paper on the webinar. With Idan. No, on the Salesforce what? No, the dense paper is 3D reconstruction of the. Maybe anyway. So this is a very big issue. Now, for Dan's question, whether there is a way to prove randomness, you're a questionnaire. So I don't have an answer for you, but I think Tali Tishki does. He's very emotional and opinionated about that. This is not, they cannot prove randomness, but he has all kinds of fancy ways
Like
to
prove beyond reasonable doubt.
Yeah. So I think Tali Tishki has some kind of. He once tried to explain to me with a lot of integrals on the board, and then I lost him halfway through. Something with fishy information. But so that's for your question. I think there are more sophisticated ways to get closer to a real.
Wait, so are you saying if all these connections are random, the money going into TAARS
going to present the paper.
Speak to the microphone. Turn the microphone.
שלום. שומעים? Okay. So, I'm going to present the paper of the one et al. That was published in Nature last year. It is called Non-Redundant Coding of Aversive Odors in the Main Olfactory Pathway. Great.
You need to connect the USB.
Yeah, I did. Okay, never mind. Okay, in the main olfactory system, there's a wide variety of olfactory receptors, which we've talked about in class. But researchers have found that in 2001 that apparently there's another group of olfactory receptors, a small one, consists of only 14 receptors, that is called the TAAR receptors. So what are TAARs? TAARs stands for trace amine associated receptors. There are G protein coupled receptors, like the receptors we've talked about in the main system, in the main receptors, and they are encoded by the TAAR genes, and they all detect amine molecules. Right? So, what are amine molecules? Amine molecules are organic compounds containing nitrogen atoms. So, they all share. And also, they all share a characteristic aversive smell, okay, like ammonia. And this is one example of ammonia, actually. For amine, here's another one, and I could continue all the lecture, but I gotta continue. Okay. Okay. The paper tries to discover how the TAARs contribute to odor perception. Okay, through genetic manipulations, of course. First, they removed all the TAAR genes from the genome. Okay, then they removed only a single TAAR gene, and we will see what happened to the behavior. And we also we will see what we may learn about the redundancy in the code from their findings. So, first, they investigated the functional consequences of removing all the TAAR genes. So they created a knockout mouse strain that lacks all the TAAR genes from the genome. It is called delta T29. And they checked the glomeruli responses. They checked the odor-evoked glomeruli responses of these mice. They did it in a very interesting method that I didn't hear about before. It's called in vivo optical imaging through synaptofluorine. So this method makes use of the low, the acidic pH level inside vesicles, and okay. So synaptofluorine is a pH-sensitive molecule. It's a pH-sensitive molecule, green fluorescent. It's a green fluorescent protein. It is genetically encoded, and it's genetically encoded indicator of transmitter release. There, the pH is very acidic, as we know. So, when the synaptofluorine is in a very acidic pH level, it is non-fluorescent. It's non-fluorescent. But when the vesicles are released, so it's exposed to the neutral extracellular space, and the presynaptic terminal becomes brightly fluorescent. Okay, so as we know, glomeruli receive inputs from sensory neurons that express the same receptor. We saw this figure already, and we expect that if we remove the TAAR genes from the receptors, if we don't have receptors of TAARs, we will not have a glomerulus. The corresponding glomerulus will not respond. Okay, so in accordance of previous observations, they used 5 different amines and low concentrations of the amines, and in the wild type mice, they saw that there is a response. There is response in the amines for the amines, and also glomeruli responded for non-amines. Okay, but in our delta T29 mice, we don't have these responses to amines, but the responses to non-amines persisted. Okay, so we can conclude that all of the high sensitivity amine responses derive from TAAR glomeruli. Okay. The non-amines? It shouldn't be, but
It's in the same.
Yeah.
It's in the same animal.
Because
these are wild type and these are delta T29. So yeah, it could be a variation in the response. Okay, so what about natural stimuli? Urine is a rich source of amines, especially with PEA, and they wanted to see whether they will got a response in natural stimuli. It's a predator urine. So we can see that, okay, I forgot to say that we know that urine, the TAAR4 specifically is responsive to PEA and to urine. So in the arrows here, we can see the TAAR4 glomeruli, and we see that they're fluorescent, but here in the non-amines, other glomeruli are fluorescent. Okay, and as we expected in the delta T29, there are no responses for the urine. Okay, any questions? So we can conclude that the most sensitive urine responsive glomeruli correspond to the TAARs. Okay. So what about behavior? Okay, do the TAARs mediate the aversion? Okay, so they performed the behavioral experiment. Wild type, heterozygous, and homozygous delta T29 were tested in a 2-chamber preference assay, where the mouse could occupy an odorized or a non-odorized or an odorized compartment of the cage. They used heterozygous delta T29 as a control because the knockout heterozygous they have one wild type allele, right? So they either respond like the wild type mice, or they have a reduced effect is observed in these mice.
Did they check how many receptors are expressed in the heterozygous mouse?
What do you mean?
They could check if the expression of TAAR receptors is similar to wild type.
Actually, not in this paper. In previous papers, they saw that heterozygous most of the time have some cells express the wild type gene, and some cells express the TAAR gene. So
there are TAAR receptors, but less.
Yeah. Yes. Okay. So we expect here reduced effect. Okay. So these are the results. In the x-axis, we can see the amines and the dilutions of the amines. The percentage here is the ratio of the amine inside a water solution. And the y-axis here is the aversion index that is calculated by the time that the mice spent inside the odor compartment minus the time the mice spent in when they administered a water rather than the odor to the compartment. So we can see that all the bluish here, the controls, the bluish columns, the control mice, did exhibit aversion from the amines. Okay. So I just wanted to say that negative aversion index represents aversion from the amines. All right. So we can see that the controls exhibit aversion, but in low concentrations of the amines, the homozygous mice don't perform, didn't avoid the odorized compartment. So we see that in low concentration, the responses are TAAR dependent. The response is TAAR dependent only. Yeah.
But wouldn't we expect a zero aversion index for the complete knockout? So it seems to be significantly different from zero in the other direction. Is there any explanation for that?
Actually, they didn't say anything about it. I also thought about it, but I didn't have a good explanation for this. Yeah. Okay. And we see that in the high concentrations, when the concentrations are too high, the more receptors come into play, and they avoided also from this chamber. But what about the urine stimuli? When they introduced a puma's urine, they see also that the mice didn't avoided the chamber. So removing all the TAAR genes abolishes the high sensitivity to amines. Okay. Okay, but now we want to see what happens when if we remove only a single TAAR gene from the genome. Okay, so they created a delta T4 mouse without the TAAR4 receptor, and we said that the TAAR4 responds selectively to PEA, okay, and to predator urine, and we see that we don't get a response in the knockout mice for the PEA, but the response for IPA is still there. So, okay. But what about the behavior? So you know how to read this figure already, and you see that also they didn't avoid the PEA and the predator urine, but the aversion index for the other amines is like the controls. So if we remove a single TAAR gene, it abolishes the aversion to certain amines. Okay? Good. So let's summarize our results. We saw that the TAAR contributes significantly to the perception of amines. Okay? We saw it by removing all the TAAR genes. It abolished the high sensitivity to amines. Okay? And we And removing a single TAAR gene also abolishes aversion to certain amines. Okay, so okay. So also, we saw with Yoram Ben Shaul that the verbonasal system is the verbonasal system mediates aversion from predators by direct contact with their urine. So this system, the TAARS system, may contribute to predator avoidance over longer distances because they are responsive. They are sensitive to urinary volatiles. Okay, and also we saw that the aversive quality is encoded in a non-redundant manner. What do I mean? Okay, it is generally thought that that auto representation is very redundant and distributed. Okay, so each glomerulus contributes a small contribution to the representation of a given odor. Okay, okay, but so in this view, if we a deletion of only one glomerulus will result in the same behavior of the animal. But now here we saw that when we moved only one glomerulus, we got a different behavior of the mouse. So it means that we have that it is the code is less redundant than previously thought. Okay, this could explain also the fact that vertebrates retain a lot of the olfactory genes in their genome. Okay. Okay, so actually, I think that this paper was really well written and very straightforward. They used molecular tools and behavioral tools to get to their conclusion. But some comments I would like to make. First, can we assume non-redundancy in the code based on one single TAAR receptor? It's one receptor from a secondary type of receptors. To conclude this strong conclusion just from this is a bit far-fetched, and they could also play with more TAAR genes or more olfactory receptor genes to support their conclusion. Also, they examined only one behavior. Maybe for different amine-sensitive behaviors, the code will be different. They looked only on aversion from predator urine, but what about mice urine? I don't know. Maybe a social behavior will be elicited, and different type of code will be. We'll see different type of code. Okay, now a small comment. In the removing all TAARS behavioral experiment, they could use more type of predator urine to get more convincing results. What I refer to this figure, we saw that the standard deviation here in the puma's urine is also in the aversion side, and we know that they have more urine in the freezer of lynx because we saw in other figure that they have this urine. So why not using it here also? And that's it. Questions?
Can you explain a little bit more about why the vertebrates would retain the genome? You mentioned that the aversive quality is coded in a non-redundant manner. How does that relate to the genome?
Okay, we know that we have like 20,000 genes in the genome, and we have 1,000 genes that are just for olfactory receptors. Okay, so it makes sense that we should retain more genes, this 1,000 genes in our genome, to if the code is non-redundant, to be able to sense these all the space, all this, I don't know, the space domain of the orders that we can. And if the code was more redundant, we just, I don't know, maybe 100 olfactory receptors could encode all the others. Okay? Just by different patterns of activity.
Right.
Okay?
So you're saying that there's less redundancy but more space instead of more space but more redundancy but less space.
Less redundant. What do you mean?
There's less redundancy but a larger space of cells that you can
Yeah.
smell.
Yeah.
That's kept from generation to generation. Do they talk about that at all?
No,
Or is
actually.
that's something you can...
They mentioned this comment, but they didn't elaborate on it more.
You mentioned that they could check different targets.
Yeah.
And in the last slide, you showed that they removed they knocked out one TAAR
Four. Yeah.
They didn't try to remove the other?
No, not in this paper. In another paper, they saw they figured out that the TAAR5 has its role in social behavior, but they didn't. In this paper, they didn't eliminate any other gene.
But they did had mentioned at the beginning that all of the TAAR genes, they didn't express all of the TAAR genes.
Yeah.
Just a small remark to what you said as a response to Lily's question. I think there is a difference between whether a code is redundant or whether a code is distributed. I think what you explained should be more called a distributed code. It means that the activity of several receptors together gives the information. But this has not so much to do with redundancy, meaning that if you delete out some of the receptors, you still have a good coding of the stimulus.
I agree that there is a difference between them, but I think that they are very correlated. They have some things in common, because if you have distributed if you have a distributed code, so I guess that just by deletion of one glomerulus, you will get a different type of activity. So
Actually, there could be a very long discussion now.
okay. Thank you.
כל הקונקשן,
כן.
זה
יופי, מה זה?
ממש אני מסכים.
כאילו, התקשרת נעליים.
אל תגידו לי אם זה חשוב.
שומעת?
לא,
זה זה?
לא,
לא, אבל זה באמת זה עושה שם, כי זה לא...
גם
זה
במוזיאון
זה מתחבר, זה נורא מעצבן. זה לדעתי זה נראה לי כמו מהצ'קסים, זה פה לא...
זה אלף סטאטר.
נורא מעצבן לי אבל, שאני... יש לי מלא דברים להצביע עליהם בטקסט. בחיים שלא השתמשתי באנטלנט.
אולי גם ביד.
אני בדרך כלל ביד, אבל זה לא חינם. כאילו, תסתכל, כאילו... אני צריכה עכשיו להצביע, כאילו...
פקטורי בר.
אני קצת... אוי.
אלה.
כאילו... כזה... אבל רגע, אתה אומר שזה זה...
הוא נכון גם
כל הכבוד.
רק שלא יעשו לך יותר טוב.
טוב. רציתי להוסיף.
האם, אתה יודע, תמיד אתה בלאר פור מקבל כך וכך דינר סוף בלאר סוף בלי, אתה לרוס, כמה צוותים ניגשים וכמה ניגשות, אבל אנשים מאוד אמושיונל.
stimulation by a network of electrically coupled inhibitor interneurons. It was written by Zhu, Frank, and Friedrich, and it was published in Nature a few months ago. So we start by looking at the olfactory bulb. So we all know that the input to the olfactory bulb is coming from the olfactory receptor to the glomeruli level, and we treat the mitral cell at the output level, and the axon of the mitral cell send their axons to the brain. Usually, when we ask questions in the olfactory bulb, we observe a certain input-output function, and we ask what is the computation that's going on between. So first, I want us to focus at the input-output function in our case. So the function is intensity invariant activity, which means that at the input level, when we give different intensity of stimulus, there is high sensitivity to intensity. For example, if these are different glomeruli, so this is the response for weak intensity of another, and this is the response for strong intensity of another. There's high sensitivity. On the other hand, at the output level, at the mitral cells, the sensitivity is decreased, and previous studies show that activity patterns for different concentration of odors are not decorrelated. And also, if you think about it, at the higher level, the perception level, the perceived quality of odor is stable through a range of concentration. So this leads us to the hypothesis that mitral cell activity patterns are equalized by interneurons. So from now on, we will focus on the computation. So what they suggested in the paper, that this computation is done by interneurons in the olfactory bulb, and they create the intensity invariant activity which we saw.
Just intensity, does it mean like only concentration of odorants or
Concentration of odorants.
concentration?
Okay, so in order to begin, I wanted to understand the computation in a better way. So we look at a very simple model. Let's say we have n input neurons and 2 output neurons that just sum linearly the input of the neuron. So in this case, we have an input, an intermediate level. We see that the left neuron passes the threshold and fire because all its inputs are activated, and the right neuron doesn't pass the threshold and doesn't fire, and therefore we get the correct classification. And let's now look what happens when there is weak intensity. So the input is not enough for the left neuron to pass the threshold, and therefore it doesn't fire, and we don't get a correct classification. And when the stimulus is very strong, the right neuron also passes the threshold, and we get false positive. So it means that when there is high sensitivity to the intensity, it creates problem for the classifier. A way to solve this can be by a simple normalization of the mean activity. For example, if we divide the activity by its mean, we get kind of an average response independent of the intensity of the stimulus. Unfortunately, in our case, the problem is harder because when the intensity is increased, not only the magnitude of the response is increased, but also additional glomeruli are recruited. So not only the mean of the activity is changed at the glomeruli level, but also the distribution of the activity. So this is another property. And in this case, simple normalization won't solve the problem. So what we are looking for the computation, we are looking for a computation that equalizes both the mean and the distribution of the activity at the glomeruli level. So now that we understand the computation, we can dive into the paper. So what they did in the paper, the animal model was the zebrafish. The zebrafish is good for studying the olfactory bulb because its olfactory bulb is similar to that of larger vertebrates, and on the other hand, it contains a relatively small number of glomeruli. And this is a very beautiful imaging of the olfactory bulb of the zebrafish. So what they did in the paper, they genetically targeted a subpopulation of GABAergic interneurons, which are called DLX46 neurons. And remember them because we're going to talk about them in the next 20 minutes. What they did is they showed how the DLX neurons modulate the output of the mitral cell, and they tried and succeeded to explain what are the underlying mechanisms. So what we'll do is we'll follow the process step by step. So the first thing to do is to see what kind of we get at the mitral cell when we activate the DLX neurons. So what they did is they activated the DLX neurons using optogenetics and measured which kind of currents they get at the mitral cell. So what we see here is that they got two types of currents. Under halting potential of minus 70 millivolts, they got an inward current, which is an excitatory current, and when they hold the cell under higher voltage, they got an outward current, which is an inhibitory current. So the next step was to see what causes each of these currents. So they try to block the current. So here what we see is they try to block, we know that these are GABAergic interneurons, so they try to block GABA release. So what we see here is that, let's first look at the gray. This is the control. This is the firing rate of the mitral cell. So here we see that the mitral cell is firing, and when the optical stimulation is on, we see that the mitral cell stops firing. So when we activate the DLX neurons, they inhibit the firing of the mitral cell. However, when we block the synapse, we see that this is the black. We see that the mitral cell didn't stop firing, meaning we block the inhibition when we block the GABA release. So this is the inhibition. But when we move to the excitation, they couldn't block it using a chemical synapse blocker. They could block it only when they use gap junction blocker. You can see it here that when they use gap junction blocker, the current becomes from this to this. So we understand that there are two types of connections. There are both chemical connections, which involve GABA release and cause inhibition of the mitral cells, and there are electrical connections, which involve gap junction and cause excitation of the mitral cells. So we understand what are the connections between these neurons. And now the next thing is to see, the interesting part, to see what happens during an auto response. So what they did, in order to learn the function of the DLX neuron during an auto response, they inhibited them and tried to deduce what is the function. So they inhibited the DLX46 neuron using optogenetics. They estimated the firing rate of the mitral cell, and now they try to see what happens when we inhibit it. So what happens when we block the current under different concentration of another route, and they block both the excitatory and the inhibitory current. So now we see some interesting results they got. So just attention. It's a little bit confusing because we're looking at the inhibition of these neurons, so we can deduce the opposite of the effect we see. So just a reminder for me. So what we see here is three different concentrations. This is the low concentration. The different rows are different mitral cells, and this is the firing rate, and the axis is the time, and this is when the odor is applied. And what we see here is that for low concentration, if we compare the control condition to the condition where we when we inhibit the DLX neurons, we see that for example, look here, these cells, their firing rate is decreased. This is low concentration, the firing rate is decreased. From this, we can deduce that the DLX neurons increase the response for weak stimuli, because here the response is reduced. On the other hand, if we look at the strong stimuli here, we see that the firing rate of, for example, look at these cells, their firing rate is increased when we inhibit the DLX neuron. So total, we can deduce that the responses to strong stimuli are attenuated. So responses to strong stimuli are attenuated, responses to weak stimuli are boosted. And if you remember, this is what we wanted when we looked before. This is a kind of a way to solve the intensity invariance in the manner of the mean activity. And now, just to see if you are with me, so we can also understand, they performed another step and understand what causes each of these effects. So responses to strong stimuli are attenuated. So if they are attenuated, what can cause this attenuation? Anybody have an idea? Attenuation is a result of inhibition. So what causes the inhibition? No, it's the same interneuron. There are electrical connection and chemical connection. So the chemical connection involves GABA release, so this causes an inhibition. So spikes of the DLX-first neurons cause attenuation of the strong stimuli. On the other hand, the boosting of the weak stimuli is caused by the excitation, which is a result of gap because this is what they showed, that only when they blocked the gap junction, they got this current.
Okay, did they provide a functional explanation why this
It's just that the depolarizing of the DLX neurons is passed through gap junction to the microcell. Okay? Okay, so the next interesting thing that they showed is that we talked about the distribution. So this is the control condition, and again the rows are different master cells, and now the x-axis is different concentration. So what we see here is that under the control condition, when the concentration is increased, the distribution of the activity of different master cells is not changed at all. On the other hand, if we look here at when the DLX neurons are inhibited, we see that when the concentration is increased, the distribution is changed a lot. It's much broader. So we can deduce from this that the DLX neurons also stabilize the distribution of the micro cell responses. And again, this is what we wanted. The second property we wanted an equalization of the distribution when there are different concentrations. Okay. The next thing they do is they try to suggest a mechanism that explains this bidirectional equalization, the attenuation of the strong stimuli and the boosting of the weak stimuli. So the first mechanism they suggested is called DRC. It's dynamic range compression. According to this mechanism, the input from the DLX neuron to the master cell is excitatory when the intensity is low, and inhibitory when the intensity is stronger. We can look at here, this is the input-output function. So in gray, this is what before the DRC. And after the DRC, we see that we get the black curve. So the weak stimuli are boosted and the strong stimuli are attenuated. So this is the first mechanism they suggested. The second mechanism they suggested is DRS. It's a diversification of response sensitivity. According to this mechanism, so these are the curves, in gray you see the curves of different microcells, and each cell is changing in a different way. The sensitivity of each cell changes differently, and we get the same result of the black curve. So, and it's important to mention that these two mechanisms are not mutually exclusive. What we're going to do is see whether the first one exists, and then see if we need the second one, and whether it exists too. So, first we check the DRC. So they show that there is DRC. They showed it by looking at the inhibitory and excitatory current as a function of the activity of the DLX neuron. So this is the amount of the activity, and we see that when the activity is low, the excitatory current is stronger than the inhibitory current. But then they switch, and the inhibitory current becomes stronger than the excitatory current. And this exactly explains what we wanted from the DRC, that at low intensity there is boosting, and strong intensity there is an attenuation. And if you think about it, we can even think what causes it, because the inhibitory, we see that when the activity is large, the inhibitory current is stronger than the excitatory current. And we said that the inhibitory current is caused by GABA release, by spikes. So in order for DLX neurons to spike, they have to pass the threshold. So this can happen only when the DLX neurons are activated enough in order to pass the threshold. There is enough depolarization. However, here at low intensities, there is activation of the gap junction, and here we don't need to pass the threshold. So this can happen also in a sub-threshold regime. So here we get most of the activity of the gap junction, and here there's involvement of the spike. So this can be an explanation for this. So the question now is whether this is enough, whether the DRC explains everything we see. So the answer is no. There are effects which cannot be accounted for the DRC mechanism. In order to understand these effects, we need to look at the distribution of the change index value. So what is the change index value? The change index value is calculated for each maternal cells and for each odor, and it calculates the difference of the response between when there is a manipulation, which is inhibition of the DLX neurons, and comparing to the control condition. Meaning, if this specific maternal cell to this specific odor fires more when the DLX neurons are inhibited, the change index will be positive. And if it fires less, it will be negative. So what we see here, we're looking at two different concentrations. Here is the low concentration. What we see here is the distribution of the change index values, and we see that under the manipulation, the inhibition of the DLAC neurons, most of the change index values are negative. Meaning, in this case, we already know that most of the cells fired less. The activity was reduced because the function of the DLAC neurons is to increase the activity when the stimulus is weak. But here they are uninhibited, so compared to the control, the activity is decreased. So this we can explain by the mechanism we saw. And exactly the same explanation holds here. We see that when there's high concentration, the change indexes are positive. But there is an additional effect here which we cannot explain from the DLC alone, and which is we see that under the inhibition, the distribution got broader. So, and this we cannot explain by DLC because according to DLC, the distribution should just shift, and we have an additional effect. So they try to understand, to explain this, and they saw that they can explain this effect by the DRS mechanism. What they did is they look at the standard deviation of specific mitral cells. If we look, for example, here, we take a specific mitral cell, and we take its response for different orders, and we calculated the differences in the change index, which is the difference between the inhibition and without the inhibition. So, for example, these are the change index of specific mitral cells, and we look at the standard deviation of this cell. What it means, if the standard deviation is very high, we can say that this cell responds very different, the effect of the DLX neurons on this cell is very different to different orders. There's a big difference, the high heterogeneity. On the other hand, if the standard deviation of this cell will be very low, we can say that the effect of the DLX neurons on this cell is independent of the stimulus, independent of the identity of the order. So we can see the results here in this graph. We see that most standard deviation, this is the standard deviation values of all the individual mitral cells, and we see that they are very low compared to what? We compare that at the gray bar, you see the standard deviation of the population, which we just showed that was very large under the inhibition. So from this we can deduce that the variability of manipulation effects on responses of individual mitral cells to different orders is much smaller than the variability of the effects across the population. And from this we can deduce that the effect of the individual mitral cell is systematic, meaning the DRS holds. It means that the DLX neurons changes in sensitivity of different mitral cells in different directions. They also showed, and I thought I won't have time, but I have, they also showed that the tuning curve of the mitral cells is not changed a lot when the DLX neurons are activated. Meaning it's not a matter of identity, it's a matter of the intensity of the order. This is the conclusion. So I was very fast. We can summarize. The DLX 46 neuron equalize both the mean and the distribution of activity across the mitral cell population. The two mechanisms we saw, the DRC and the DRS, extend the input range of mitral cells and they enable the order representation across mitral cells become robust against variations in stimulus intensity. And the DLX 46 neuron modulate mitral cell responses as a function of stimulus intensity rather than identity. And this is what we want to show. And last, just my personal point of view, it was very challenging for me to understand this paper. They did a lot of things, and it was also challenging to bring it here. And what I really liked is that they suggested very high-level mechanism of the DRC and the DRS, and that if you look at them, it seems it's hard to understand them, but then they explained everything to the finest detail, to the physiological property of the cell, and I liked it. And also it was like a complete process of asking questions, raising hypotheses, checking all the steps, and then they answer, and I didn't see any hole in the left, and so it was nice. And it is mine, I don't know. That's it. And do you have questions? It's called glowfish, and they insert some kind of protein, and they use them there. Question.
the receptors themselves produce different output according to stimulus intensity. So the whole process of equalization is only necessary if from the receptors um, the signals that arrive are modulated by stimulus intensity.
I didn't understand the last part of it.
If the receptors don't produce output that correlates with intensity, then this whole equalization is not necessary.
Yeah, but they do. I mean, it's a known fact that you have different activation at the glomerular level depending on there's high sensitivity to the stimulus intensity at the glomerular level.
And
High sensitivity.
but did they? Okay, this is known. But did they check whether the amount of modulation at the glomerulus level is in the same range than the capabilities of this equalization system? So if
I'm not sure if I'm not sure if I understand exactly, so I answer you and tell me if I understood you. At the glomerular level, there's decorrelation between different intensities. Totally decorrelation, and they're not correlated. Okay, so I mean I'm not sure I understood you.
so it's possible that the input to this system in nature varies much more than they checked in the experiment, or it's also possible that in nature it doesn't vary that much, and that the experimental manipulations they made are out of the range of what is going on in everyday.
I didn't check if the concentration they use are physiological. I think maybe I didn't know. I assume yes, because otherwise.
Yeah, normally those are special amino acids that they use in fish in the water, and normally those are ranges that fish can react to them in nature, or that you can find them in nature. They don't have very good bioassays in zebrafish. Actually, very poor behavioral assays in fish, but those are the. This is because they can't learn. They're rather stupid animals. They can learn. I actually saw a seminar a few weeks ago that you can teach a zebrafish to avoid heat to their eyes, directly to their eyes. But yeah, no, they flick their tail to the left, and if you can teach them to flick their tails only to the right and not both left and right,
Okay.
to avoid a nasty stimulus.
So naturally, the environment hits to the eyes. Something that they're supposed to know, right?
Anyway, that's
Algebraic homework.
that's in terms of the behavior. But the physiological range of the other stimuli here is physiological.
Okay.
Yes.
What about how does the model compensate for attention or for aversive stimuli? So for something you have to pay attention to, the microcells can't just be averaged out, right? They have to have a stronger output. Whereas the interneurons here, they're trying to stabilize all the different types of inputs. So how does the model account for more interesting stimuli that they want the brain to pay more attention to, I guess?
The animal was not alive, so attention, and it's a fish. They didn't consider this property. They looked only at the output of the macrocell as it is.
And that's it.
Yeah.
Is this something that's done in other parts of research to compare,
But
to see how the interneurons react to a stimulus that requires more attention?
what do you mean by stimulus that requires more attention?
So for example, if I'm in this room and I smell all sorts of different things, fine, but if I smell gas, something tells me, oh, there's gas. So my microcells will probably have a higher output to gas or to noxious stimuli, basically.
It
So
might
how
be,
does-
but it might be the perception.
Right.
You mean top-down modulation or intrinsic or innate
Right, some kind of
sensitivity?
top-down modulation. So in the previous paper, in Amir's paper, it looked at the TAAR receptors, right? So how would interneurons of those microcells modulate the smell of the urine, for example?
The encrypt the identity of specific glomeruli receptors.
Right.
I think that's
But it's possible that attention happens at a much higher level.
yeah. I would think
So the,
like Michael said, it's a top down. I would think, but I don't know.
So actually, I think it's a good point because one paper, Amir's paper, shows us a very labeled line, or at least at low intensity, there is an input to a given line that induces the full behavior, and that's it. And on the other hand, Rainer, so that's the genetics group people, and those in their assays and behaviors, there's no computation is going on. It's an on-off thing, no interneurons. It's a very binary, very simple way to operate with the nervous system. Rainer is on the other extreme, very computational neuroscientist, thinks that a neural network is doing some computation, and the different neurons in the network are responsible for this computation. What exactly is this computation? Is it the DRC or a DRS or whatever? And in that context, I think Lily's question is very good because people are very much into how will the mitral cells or any other projection neuron network will change their firing pattern with attention, and it's very much modulated from top-down processes. Well, factor about 2 has a lot of non-modulatory input coming in. This is also something which is very interesting to the field, but we are still not there in terms of how does what is the mechanism, because a lot of time these non-modulatory inputs are like soups. They're spilled in the neural tissue, and it depends, which interneurons express which receptors, and to what extent they will be modulated and affect these processes. So I think you saw both sides of the strategies of looking into neural circuits. One is a very clear-cut Drosophila-like system, labeled line, and the other one is, lots of very fancy computations are going on in the network, and if we learn in the ICNC, we will be able to calculate what the neurons are calculating. Bon. Okay. Thank you very much. This completes the olfactory system. Last will be the auditory system.